Welcome. This is Dr. Michio Kaku, professor of theoretical physics at the City College and the Graduate Center of the City University of New York, and this is Exploration. Every week on Exploration, we discuss the fascinating world of science and its impact on society. And today, we're going to bring on a very special guest, a world leader in genomics. We're going to be talking about the Human Genome Project. We're going to be talking about viruses. We're going to be talking about HIV, cancer, gene therapy. We're going to be talking about this revolution in biotechnology and medicine. We should also point out that Dr. Frank Ryan is a consultant physician at the University of Sheffield in the United Kingdom. And we were very privileged to be able to hook up a radio interview with him all the way to England. He's the author of a number of great books, including The Mysterious World of the Human Genome. He's one of the world's authorities on viruses, and he's written a book called Coevolution as well. And so once again, today in Exploration, we're going to be talking about medicine. We're going to be talking about the Human Genome Project. Have you seen all these advertisements on TV saying that you can analyze your DNA and find out where your ancestors came from hundreds of years ago? Is that really true? Or perhaps you've read in the New York Times and other journals that there's a new cancer therapy which is shaking up the whole field using gene therapy, that is modifying the immune cells of the human body, modifying them, and then putting them back in the human body so they identify cancer cells. This could be a game changer with regards to cancer therapy. And so all these discoveries, we're going to be talking with Dr. Frank Ryan. He's a consultant physician at the University of Sheffield in the United Kingdom, where we were able to hook up an interview. And he's the author of a new book called The Mysterious World of the Human Genome. The first question for you, Dr. Ryan, is how did you as a youth first get interested in science?
I think it's because I was mathematical. I was very mathematical. It's the only subject in which I've never been beaten in an exam.
Uh-huh.
Sometimes come bottom of the class in other subjects, but in maths I was always top. And I think because I have that kind of pragmatic and analytical type of approach, the sciences were more attractive to me. But I still love the arts. Most of the scientists I know actually do something in the arts, and I love literature, I love music, and so on. So I'm not too orientated. I'm not totally orientated towards science.
Then what steered you in the direction of medicine, and in particular, genetics?
Now you're going to come across a whole series of serendipities. I actually started off with engineering, which is what you'd expect with this interest in mathematics. But I had a life-threatening motorcycle accident.
Oh.
Ended up in hospital, and I saw medicine. I never even thought about it. But I looked at it and I thought, my goodness, this is interesting. 15 months later, I entered the University of Sheffield to study medicine.
So that motorcycle accident was really a game changer. It was a life-changing event. So in particular now, what steered you in the direction of genetics?
Yes. Well, we were taught genetics, of course, as part of the standard kind of medical curriculum. But it was fairly new. I mean, this is quite a few years ago. And so, for a start, I was just interested. As soon as I had learned a little bit about genetics, I just found it extremely interesting. But actually, the way in which I then got into it was because of a related interest, which was in microbiology, and particularly in viruses. And I actually did my first viral experiment as a med student.
Is that right?
I was supported by the Wellcome. I got a grant from the Wellcome Institute to do studies on immunity in rabbits using a phage virus. This is a virus that normally infects bacteria, but you can inject it into rabbits, and it causes no disease, but their immune systems respond just as if they've been invaded by a plague virus. And I studied what happened within the immune system of the rabbit when a virus entered the bloodstream. Later on, as a doctor, I did exactly the same experiment on myself. And people,
is that right?
And I found something very strange, because for a while, I was going to become a microbiologist. That's where I was heading. But I was told, oh, no, we've got antibiotics and got vaccines. It's a waste of time, which was completely wrong. advice I was given at the time. And I began to move over towards gastroenterology and general medicine, dealing with medical emergencies and all that. But actually, in my research interest, I continued with viruses and all this sort of thing. And I discovered that people suffering from ulcerative colitis, the spleens were damaged, and it was actually threatening their lives sometimes when they went for operation. And when we actually looked at the spleens, in some patients, the spleens were only the size of your thumb. It's a condition called hyposplenism, and it fitted exactly with the sort of research I've been doing with rabbits. So I tested the patients and me and normal controls with the same bacteriophage virus, 5X174. And in the patients who had hyposplenism, they didn't respond. So you can see there was something very wrong with their ability to respond to a particular antigen arriving into the bloodstream. So this is a new thing. And then, lo and behold, colleagues who are working in other disciplines involved with other autoimmune diseases found this actually, hyposplenism, was a feature of the autoimmune diseases. Now, the other thing I did is I did blood antibodies. I did a blood antibody screen in my patients, but looking for all the other autoimmune diseases. And I found that patients presenting with ulcerative colitis actually had autoantibodies that you find in some of the other autoimmune diseases. Not every patient, but with a very significant kind of number who did have this. But when I followed them up for 10 years, some of them developed the other autoimmune diseases. So in other words, the autoimmune conditions are all linked in some way. I think that was a very important observation at that time.
I see. Well, let's now talk at a rather elementary level about genetics. First of all, genes. How many genes are there in the human genome, and what do genes do, for example?
That's not a good question, because before the human genome, the kind of first rough outline of the human genome was arrived at in the year 2001 with a great hoo-ha. And we were expecting that we thought there were about 80 to 100,000 proteins in the human body, so we expected that there would be 80 to 100,000 genes. But when the genome was evaluated, they found that there were far fewer, and now we know, because of research done by colleagues of mine in Sweden, that there are 20,400 genes, and yet there's code for 80 to maybe 100,000 proteins. And that was one of the big surprises, and we didn't quite understand it when the human genome was first evaluated. But now we know that those genes aren't just a single linear thing that just pops out and codes for a protein. The genes are made up of exons, which are like little stretches within the gene, and they're separated by stretches called introns, and the only parts that are expressed as proteins are the exons. Now, you could have a gene with, say, 8 exons in it, but when it codes for a protein, a certain protein that might just take 3 of those exons, and then a different protein might be 2 others or 3 others. In other words, that's called splicing now, and as a result of splicing, 20,400 genes could indeed code for 80,000 or 100,000 proteins. Well, there was something else that was really very odd about this. Excuse me. If we looked, I sent you a little pie chart of the human genome as we see it now, and if you look at the actual portion of our DNA, that's a pie chart of our DNA and what it is, and only 1.5% of our total DNA codes for protein. So what's the rest of it doing?
Some of it is junk DNA, right?
There is junk DNA, but I warn students and other colleagues to be very careful, because people keep deciding this is junk. We don't know what that does. And if it's viral, which is one of the things we'll come to, they say, oh, that must be junk. Like it's the relics of past retroviral infections. And they couldn't be more wrong. Those viral infections are extremely active. Not all of them, but a lot of them are extremely active. We could talk about that a little bit later. But at the time when they did the 2001 genome, the other thing that was really strange is that they didn't measure DNA directly. They measured messenger RNA. In other words, the RNA that was coded for by the DNA. And when they did that, half of the pie chart was blank. They didn't know what it was doing. So what do you think they called it? Junk.
Right.
Now the ENCODE project, which was set up to look at what they called junk, has decided that at least maybe, it is a bit controversial. Some people do disagree with this, so I better say that. But the ENCODE people doing that now think that at least 80% of that, which was said to be junk, is not junk, and it's involved in the epigenetic system. And that's a very, very interesting system, as we'll come to.
Okay, we'll get into that in a moment. Now, in the United States, we have a lot of advertisements concerning, personal genomics and the ability to trace your ancestral roots. And there's so many advertisements saying that you can find out that you're 30% British or 40% German or what have you. And the question is, well, how accurate are these things that we see heavily advertised now on TV that say that you can trace your ancestry?
Well, I think it depends just how extensive the testing they actually do. If extensive, and I suppose the more extensive ones are more expensive, naturally. But if they did this extensively, and the answer is yes, it's because the history of human migration is actually written into the genome. It's written in a very curious way. Imagine at some time there was a population, say, people believe that modern humans moved into Eurasian landmass from Africa. That's slightly controversial, but not very. I think most people believe that now. As they moved and spread out over the landmass of Eurasia, when they reached certain points in that, the mutations took place in the genome, basically. And these are called founder mutations. Now, for that mutation to matter from this type of tracing ancestry, which is haplotyped, they call it, what you get is a mutation that doesn't matter. In other words, it's a mutation in part of the DNA that isn't a key area for coding for a protein. That's the first thing. The second thing is it's also a very small mutation in an area that isn't broken up by what we call sexual homologous recombination. That's the kind of mixing up you get when you're forming the germ cells. Now over time, that mutation is passed down to all of the descendants of that single individual that had that mutation. And over time, as their descendants moved about over the planet, more mutations took place in the vicinity of the original one. So by, within about 100,000 years, you probably have 3 or maybe 4 mutations, all very closely together, very specific in that region. Now, if you look at a person's genome, they're looking for those types of mutations, and they know now by back tracing whereabouts people were or who they were when those mutations took place. They can trace very accurately where your ancestors came from. One of the fantastic things recently was they dug up this king, Richard III.
Mm-hmm.
He was buried. They found him under a car park in Leicester.
Right, I read about that.
And but people wondered, was he really Richard III? And they chased that up with some relatives in Canada, I think, and confirmed that he was. But they also looked at other things, like Native Americans, for example. When they looked at the Native American genomes looking for these haplotypes, they found that the haplotypes of Native Americans are very much those of sort of Eastern Asia, Northeast Asia, and they can absolutely trace them with a considerable accuracy to exactly where they came from. And they're all very closely related to each other.
Amazing. And isn't it also true that when they looked at Polynesians, there's been a great debate about where the Polynesians came from, that a lot of them came from Taiwan, of all places, and that it's basically...
I think if they've done it, it all depends how certain they are about the haplotypes. Sometimes it's a bit dubious. Where they get a really strong trace and can have followed it through whole lines of people and so on. Whereas that's certain, if that's what they've done, then I think it'll prove to be correct. And we are discovering some tremendous surprises in all that. We could talk about Neanderthals and also this story, the DNA story, is just absolutely wonderful and yet very curious, because if you look at the structure of DNA, it's very simple. It's a wonder that you've got something with such a simple structure, just 4 code letters and a small number of other molecules that support. I did a railway kind of analysis to explain what DNA is and how it works. And it's a very, very simple analysis. And we can go through that if you want, but it's just such a simple, beautiful structure. And it's responsible for coding of heredity, all the sort of controlled areas, the regulatory regions, all that sort of thing. And all of that based on a four-letter code on linear structures. The other thing that's so astonishing that people don't realize is that we have 46 chromosomes, as I'm sure you're aware, 23 each from each parent. And they're matching chromosomes apart from the sex chromosomes, where you get two X's in women and X and Y in men, but otherwise they're the same in men and women. And these 46 chromosomes, each of the chromosomes, the DNA, is a single molecule. It's just an extremely long single molecule. So 46 molecules code for it.
Yeah, it's amazing, isn't it?
It is an amazing structure. Amazing structure.
Now, when we look at the past, it's sometimes said that Genghis Khan, the great conqueror, Genghis Khan was, quote, the father of his country. But looking at the Y chromosome, which you mentioned is not changed from generation to generation, a lot of the Mongolian people have the same Y chromosome. So some people think that therefore Genghis Khan and his descendants really are the fathers of their country. Is that right?
I think he had an awful lot of wives. So I think that he probably has given rise to an awful lot of offspring. In fact, there's a guy who lectures in evolutionary biology with me on the genome, and he's called Chan here in Sheffield, and he thinks he is Chinese, and he thinks that he did descend from Genghis Khan, at least so he tells me. So Chan came from Khan. And, I think he feels that he's and he is a geneticist, so he seems fairly sure that he is descended from it. If a guy had, say, 800 wives or something like that, and then his sons and all the family did the same sort of thing for some time, then I dare say there might be an awful lot of descendants.
Now, also, given the fact that humans are very similar to other humans, more than, let's say, two random animals like two cats or two dogs, some people think that roughly 70,000 years ago, there was an event that killed off most humans, and then only a handful of us, a few hundred, few thousand, gave rise to the entire human population. Has that theory held up?
So far, I think it has. They say there was a bottleneck, although I'd put the bottleneck a bit further back. I'd say about 120,000 years ago would be my best guess. And they think that it must have happened at round about the time, supposedly, that modern humans emigrated out of Africa into the Eurasian landmass. Now, some people thought there was a bottleneck, and that a possible cause was a volcano, a major volcanic eruption in Asia. I personally doubt that for various reasons, but something did happen at that time. And what happened at that time is there was the emergence of a specific retrovirus, a virus whose genome is now in our genome. And that virus emerged, it looks to me, at that sort of time. Now, these viruses, their way of getting into the genome is through pandemics like HIV-1. Now, if that virus really was if that virus struck those people at about that time, then you'd have a much more logical explanation of why the genome was bottlenecked at that time. For example, there's a retrovirus now spreading in the koalas in eastern Australia. It began about 100 years ago in the northern part of eastern Australia, and over about 100 years has moved down. We find that all of the koalas in northern Australia are infected. 2/3 of the ones halfway down are infected, and 1/3 in the south. In other words, it's moving south. And you can see the extraordinary efficiency of a retrovirus epidemic in the species, because it's obvious that all of the koalas in Australia are going to be infected by it. And it's been killing them in the same way that AIDS initially killed humans, so that you can see that's the sort of thing that would definitely give rise to the bottleneck.
And I understand that the AIDS virus, its genome has also been sequenced, and the family tree is also been studied. So we can also study the family tree of viruses as well, right?
Yes. Viruses are very, very unusual, very interesting. Of course, I spend a lot of time thinking about them these days. But there's something that I put forward in one of those earlier books you were talking about. It was, first of all, in a book called Virus X. And what happened is I wrote a book about tuberculosis, thinking it'll just be nice. I looked at how the cure was discovered for tuberculosis, thinking it'll sell reasonably well and all, hunky-dory. But actually, I didn't know at the time I was writing the story of the discovery of the cure that there was an epidemic of tuberculosis in New York. And when the book came out, it was extremely topical because of that. It was just serendipity again. But the book was a front-page review in the New York Times and Washington Post. Mm-hmm. And suddenly, I was a writer known to an American audience. It was just complete serendipity. I didn't plan it or anything like that. But something else happened because they had a big meeting in Boston, and I was invited across talking about this. We had Jason Robards reading sections from the TB book, and sitting next to me was the guy who was in charge, Jack Adler, was in charge of the Bureau of Tuberculosis in New York at the time. He said, "You've written a very good book about tuberculosis. Are you interested in the emerging viruses?" I said, "Yeah, well, other doctors have been saying the same to me." He said, "Yeah, I am." I said, "Well, if you are, go down to the Four Corner States now, and you'll find yourself in the middle of an all-American emerging viral plague." And I did. That changed my life. Because I was in this extraordinary position, because I was in the most developed country in the world, medically, scientifically, and all the rest. Here was a viral plague that had emerged just about 9 months before I got there. The intensive care units in New Mexico were full of plague patients, and the wards around them. And the CDC, the Special Pathogens Branch, the CDC, was heavily enmeshed with all this investigation. So I could actually watch my colleagues working, particularly the intensive care people and respiratory people. These people were suffering from all organ failure. Young people who were fit as a fiddle, dead 2 days later, drowning in their own pulmonary secretions. Then I went to CDC, and then I sat in with the guys in CDC who were liaising back with the guys I'd been interviewing in Albuquerque and so on. But the typical thing, to cut a long story short, that was this thin nombre hantavirus. And when I was discussing this with a Professor Terry Yates, who was Professor of Zoology at Albuquerque, he was one of the brightest people I've ever met. And I'd only kind of laid aside about an hour to interview him, but then he said to me that hantaviruses co-evolve with rodents. And I said, "Well, what do you mean by that?" He said, "Every rodent has got a hantavirus that co-evolves with it." I said, "Again, well, I don't quite understand what you mean by co-evolve." He said, "The coevolution is so close that if you gave me an unknown rodent," and he gave me the example of a duck-billed platypus, which, of course, is an egg-laying marsupial. It's not a rodent. But he said, let's say it was a rodent. We couldn't place it in the rodent evolution tree. If you gave me a hantavirus, I'll place it point-blank accuracy into its evolutionary niche. I was just so astonished by this that I spent time with him. I went out onto the Servietta, the big nature reserve that he was dealing with and so on. And I asked him then, after I'd done all this, I said, "Surely what you're saying suggests that maybe the virus and rodent are each changing each other's evolution." Mm-hmm. He shrugged his shoulders. That completely changed my career and life, because I came home, I said, "Who can I ask about this?" And I looked up all the authorities might be good to. Then I actually wrote to Joshua Lederberg, who was then the president of the Rockefeller University in New York, because he was an expert on symbiosis between phage viruses and bacteria. And he invited me over, and I went to see him, and he was very gracious. He allowed me a whole afternoon, which must have been difficult because he was a very busy guy. And during the interview, he said it was the toughest interview he ever had, because I'd prepared for it considerably. And then I asked him, I said, "Are viruses symbionts?" He said, "Well, they can be when it comes to microbes like bacteria, but he knew of no examples of viruses as symbionts in any more complex organisms." He said, "But I think it'd be very interesting to look into it."
Okay.
And that's what I did. He also told me to go and see Lynn Margulis, which I did. But she knew nothing about viruses.
Mm-hmm.
So I had to just start from scratch and start plugging and searching and pushing and questioning and all the rest of it. And now I think the evidence is overwhelming that viruses are the ultimate symbiote.
Mm-hmm.
And that explains the viruses in the human genome, which I didn't even know about at the time I was doing all this. But when the genome was explored, 2001, and we saw all these sequences, people are calling it junk DNA, I had very strong suspicions that junk DNA would not be an adequate description.
So really quick, all this junk DNA that people once dismissed as being just useless byproducts of random evolution, you're saying in some sense is the remnants of ancient viral infections and interactions and coevolution with them. Is that what you're saying?
It's enormously important. Now, some of it has been closed down. You'll get viral, they get the viral genome where the genetic domains within the genome have been removed, and all you've got is the regulatory regions, which are called the LTRs. But even they are heavily involved in epigenetics. And I've got a colleague who I should mention called Professor Louis Villarreal, works at UC Irvine. Louis wrote the evolutionary section of field virology, which is that massive tome, on viruses. It's the Bible on viruses for virologists. And I made contact with him because I discovered he was writing things that seemed to be saying much what I was already thinking. And actually, he, at the time, struggled to accept viruses might be symbionts because of his notion of what a symbiont was. But I redefined viral symbiosis, including a concept called aggressive symbiosis. He said once I introduced that, everything made sense. And we arrived at a complete agreement. We're co-writing a paper at the moment.
I see.
And he'd come to it from a neo-Darwinian perspective. Mm-hmm. I'd come to it from a symbiological perspective. And we're saying exactly the same.
Okay. Now, changing gears a bit, let's say a few things about cancer, okay?
Yeah.
And cancer, of course, is one of the dreaded diseases, the second greatest killer in the United States. But doesn't cancer have genetic roots?
Oh, yes. There are massive numbers of mutations in cancer. The genome is very badly damaged when a cancer is developing. So something is going wrong with the structure of the genome. You get many different mutations to start with. And equally important, not more important, equally important, you get big epigenetic changes. And in fact, if I can explain something to you about epigenetics, because it's really important, the epigenetic system basically, it encompasses all it is. Everybody thinks it's very complicated, but it isn't. You can simplify it. And you can simplify it into a number of distinct mechanisms, 4, I think, which are not necessarily dependent on DNA change. They include methylation and histone modification and so on. But basically, what these things do is they control the expression of genes. Now, if you think about it, why is a brain cell different from a blood cell? The reason why is the epigenetics are different. The actual genes, the genome, is the same. So what the epigenetic system does is it controls the profile of expression of genes, and the profile of expression is different to different cells, tissues, and so on. And you can see how important that system is.
Well, that's it for the first part of Exploration. Once again, our special guest today is one of the world's leading authorities on the human genome and viruses, Dr. Frank Ryan of the University of Sheffield in the United Kingdom. Once again, this is Dr. Michio Kaku, Professor of Theoretical Physics at the City College and the Graduate Center of the City University of New York. And stay tuned now for the second half of Exploration. Welcome. Once again, this is Dr. Michio Kaku, Professor of Theoretical Physics at the City College and the Graduate Center of the City University of New York, and this is the second half of Exploration. In the first half of Exploration, we introduced Dr. Frank Ryan. He's a consulting physician at the University of Sheffield in the United Kingdom, where we were able to track him down. He's one of the world's leading authorities on the human genome and also viruses. So we talked to him about genetics and also epigenetics. Realize that genes are not static. They simply don't sit there being fixed when you're born. No, they have to be switched on and switched off. And that takes place due to environmental cues often. So in other words, epigenetics makes genetics much richer and much more complicated because genes have to be turned on from cues from the outside environment, let's say, in order to be activated. But that also opens up a whole new world of therapies. So in the second half of Exploration, we're going to ask about cures for diseases. For example, in the New York Times recently, there was a front-page article about a new cancer therapy. Basically, you take immune cells from your own body, immune cells which cannot recognize cancer at all. You take these immune cells and modify them using gene therapy so that they have the gene which allows immune cells to recognize cancer cells and kill them. And then you reinsert these modified cells back into the body, and lo and behold, bingo, they also kill cancer cells. So in other words, the human body actually does have a mechanism to identify cancer cells, but over time, cancer cells learn how to invade that mechanism. Now with gene therapy, it's possible to rewire some of the genetic coding so that these immune cells can destroy cancer cells. Of course, there are problems. It's very costly in order to have this therapy. And also there are side effects, some rather severe side effects to this therapy. So anyway, in the second half of Exploration, we'll continue a discussion with Dr. Frank Ryan, author of the book, The Mysterious World of the Human Genome, as we explore possible cures using gene therapy and epigenetic therapy.
Two therapies are coming in at the same time, and they're very closely related to each other. One of them is gene therapy, and the other is epigenetic therapy. So we said genomic therapy, you've got them both. And I think, yes, we now understand much more about how the human genome functions than we did even 10 years ago. In fact, we understand even more than we did 5 years ago. When I was teaching the students 5 years ago, the left side of that pie chart was blank. Now that left side is filling in, and so we're beginning to understand much more. And the other thing is we've got techniques now for changing genomes. You know, you've heard of this CRISPR technology.
Mm-hmm.
Basically, I'm not going to tell you what the acronym stands for because it'll be meaningless. But basically, it's a discovery made in bacteria for bacteria to deal with viruses. You see, viruses coming into it again. Bacteria use this technique to get rid of viruses when they invade them. And basically, what CRISPR will do, and we hadn't anything like this before, and this has only come in the last 5 years, is that it has the ability. You can add a little piece of RNA which will select part of the genome you wanted to do it to. Then it goes into the genome and it'll go up to your DNA and it'll cut through it, both strands, as if scissors. You cut it with scissors. Now, normally the body would just, our own genome kind of regulatory apparatus would just join the strands back together again and repair it. But of course, if you combine that with a technique of excision or incision, you can potentially remove the damaged gene, or you could even replace the damaged gene with a new sequence. Now, that's an enormously powerful tool, and we've only had a relatively short time in sort of testing it and seeing what we can do with it. It's early, you know, gene therapy and epigenetic therapies are early, but a number of conditions are already being treated using these techniques. The results tend to vary, and I don't want to give people false hopes or anything. But if you said to me, what are the hopes for the future, and if you're talking even about the fairly recent future, the next 10 years, say, I think that more and more by way of gene therapy and epigenetic therapies are going to appear, and they're going to get better and better over time because we now have a kind of tool, or a number of tools, that enable us to do things that we were never able to do before. So it's coming. The easiest way to do it is if you can take cells out of the body. For instance, if you take bone marrow cells out of the body, then you can flatten this sledgehammer kind of treatment this, but you can destroy the bone marrow with radiation. Then you could change the cells, because if you have cells in front of you, it's much easier to change the genomes of individual cells like that, which you have in under laboratory conditions, than it is to change, say, the genomes of an organ in a living human being is much more difficult. So conditions like blood cancer, it's obvious that that's where they're going to start, and they have started. And they're already treating some types of blood cancer now in this way. And they actually treated multiple sclerosis also in a similar way. Sheffield is actually one of the centers doing it, where they take cells out of the bone marrow, irradiate the bone marrow, and then put cells back in, because the cells in the bone marrow haven't acquired the autoimmune kind of conditioning that causes multiple sclerosis. And this apparently makes an enormous difference to the patients. So these treatments are early and new. They're dangerous. There's a mortality rate. And all we can say is that they are now coming into use more and more. And I think that over the next 10 years, we're going to have a lot more of them. One of the conditions I'm very interested in is the type of dementia called C9orf72 dementia, used to be called Pick's disease. It's the second commonest cause of dementia. Now we know specifically which gene is involved, and that's the gene. Chromosome 9, the 72nd gene along it, is the gene that's abnormal. And also, interestingly, patients and families who have this condition are also prone to motor neuron disease. It's the first genetic clue we have for this awful condition. And now, obviously, I know of at least 3 different groups who are now looking to find a specific therapy because they now know what the gene is, they know what's wrong with it, they know what it's doing, and that's the first really important step for a new therapy.
Now, speaking about gene testing for diseases, recently I read an article about Alzheimer's, that there is a correlation with the APOE4 gene. It's not 100%, but it's actually caused a lot of people in the marketplace to specifically request a genomic test for the APOE4 gene, which predisposes you, but doesn't determine, predisposes you to have Alzheimer's. So you think that this kind of testing is going to become more common as we begin to identify the genetic roots?
It's already becoming more common. There's massive studies going on here in Britain and in the States and elsewhere where they're looking at something like 100,000 people. They're doing their full genome. They're following them up over a period of time. They're doing the full epigenome. They're examining them at intervals where they take blood tests, urine tests, all the rest of it. They watch whatever conditions they develop, and they're watching then to see what is it different about the ones who eventually develop diseases. Remember me with the ulcerative colitis patient. When you follow someone up long term, you learn far more from these studies, and that's what they're doing. And I think that they will be looking specifically for things like Alzheimer's disease. Now, the Alzheimer's differs from that. The only other type of dementia I spoke to you about, the frontotemporal dementia is another name for it, the one where we know exactly what's causing it in those patients with that abnormal gene. Well, the strange thing is frontotemporal dementia, we know of at least 6 specific mutated single genes can cause it. But with Alzheimer's, it's very hard to find a specific mutated gene. There is that one you mentioned, but a lot of people with Alzheimer's don't have that mutated gene. So I think that it suggests that whatever is causing Alzheimer's is perhaps more complex. One possibility might be an multiple epigenetic factors, because epigenetic change during your lifetime, where your genome, when you get identical twins, we regard them as perfect examples, for research purposes, as identicality of genome. But identical twins, by the time they've lived out their lives, their epigenomes are about 10% different. So the epigenome changes during your lifetime. And yet this same epigenome controls the expression of genes. You see what I mean?
Mm-hmm.
So it's very, very important that we moving forward, that we continue to study the genome, but also continue to study the epigenome of what that's doing to the genome. And I'm rather suspicious of Alzheimer's because we're just not finding it. It'd be bound or obvious if most cases of Alzheimer's were due to a mutated gene. I can't see how we could have missed it for so long, which suggests to me that it's more complex, maybe multifactorial, but more complex.
The next question I have is, when you talk about gene therapy, some people get a little bit nervous because perhaps if you have germline gene therapy, then your children, your grandkids, and your whole family tree will be modified just because you modified your own genome, and it makes some people nervous. But isn't that still further down the line? I mean, we're not in the situation of being able to create designer children or anything, right? But what are your thoughts?
We're not in that situation yet, but there have been 3 different experiments done in China in which they have if you want, perform genetic experiments on human embryos. Usually there were embryos either that were in some way diseased and they would never have gone to create a baby, but they have genetically engineered a human embryo. So it's obviously very trial and error. It's very, very early stage, but I have no doubt whatsoever, because again, you're dealing with just a single cell or a small cluster of cells, and it's in the laboratory. So in a laboratory, if you can get a cell into the laboratory, I think it's much easier to carry out some sort of genetic engineering. And I think that genetic engineering of that sort will come in, but it'll be introduced, I think, to treat very serious genetic disorders. We already are using a technique called pre-implantation genetic diagnosis. If, for example, a patient came to see me who had the frontotemporal dementia, there's 50% chance the children will have it because it's an autosomal dominant. Now, if they said to me, I want to have a child, what can you do for me? I'd say, well, we can offer you pre-implantation genetic diagnosis where you take the ovum, or say 5 or 6 different ova from the mother, you fertilize them outside the body, but then you wait for them to start developing into tiny little embryos, about 8 or 16 cells or whatever, and you take a cell from that, which won't do it any harm. You test the cells, and you can find of 8, roughly 4 on average will have the condition. But you can just only then put the 4 that are normal back into the mother, and not only have you prevented a very serious disease in the children, you've actually removed the condition from the pedigree of the family.
Okay, and also talking about the social implications of this. Several years ago, we had Dolly the sheep, a cloned animal, and many people said, oh my God, we're going to have cloned humans pretty soon. But that has not occurred yet, right? I mean, to my knowledge, we haven't even cloned primates, let alone a human being. But what are your thoughts about cloning? Was that overblown?
Well, I'd have to see what reason they wanted to clone.
Right.
There are enough of us on Earth already. I can't see there being a big pressure for cloning. I can see conceive a sort of science fictionish scenario where a parent lost a loved child, or losing a child from some terrible condition, and if they felt that if that child could be cloned, they'd have the child again, or something like that. It'd have to be a sort of slightly science fictionish kind of scenario, I think. Excuse me. I can't see it being a sort of medical indication, if you see what I mean.
Right.
Unless people, the other possibility is people wanting to live forever, but I can't even see that, to be honest. I think it'd be just really very far out on science fiction.
And speaking about science fiction, isn't it true that like behaviors are maybe partially influenced by genetics, but no one's ever found a gene for these things? So, for example, a shyness or boldness or something like that. So that's still in the realm of science fiction, right? Being able to isolate genes for behavior.
Yeah, I think that certainly some elements of behavior are definitely epigenetically controlled. For instance, there's a group of fish, I think a wrasse fish, where the male is highly colored and so on, and the females are dowdy sort of little yellow things. But if the male disappears, the female promptly absorbs her ovaries, develops testicles, and grows into a male.
Mm-hmm.
Which, in order to do that, the female fish have to observe the change in their circumstances, and then there has to be an epigenetic kick-in, I think, which then causes an endocrine kind of spin-off that does all that. The other thing we know is that, for instance, at the end of the war in Holland, the Germans thought they'd be ousted from Holland earlier than they actually were. There was, the kind of bridge and all that sort of stuff. But so they actually took all the food as they were leaving, and so the Dutch were starving. So it was an accidental thing because the Allies didn't actually succeed in invading as quickly as they thought they would. And so the Dutch were exposed to the starvation for longer than anyone would have thought. But that created changes in the women, in the babies that some of the pregnant women were carrying. It affected their metabolism and could be shown to change behavior and metabolism over several generations. And that was definitely conditioned through the epigenetic system. Mm-hmm. So I think you're far more likely, I think, to get, if you want, think of a child being brought up in circumstances that are very damaging. The epigenetic system of that child's genome would be affected in a major way. So when the child goes up, their behavior would change, I think. And I think not only will their behavior change, but the behavior of the children could even be changed, because the epigenetic system, there are things called epigenetic inheritance systems, and you can inherit patterns of epigenetic change that could go on for several generations. So it's a very strange and very, very, very powerful system, and it's so finite, so minute. There's a tiny little area on one of the chromosomes with a single enzyme over a very short stretch of chromosomes, histone modification thing. And if that's triggered, a change, if you just add a tiny little chemical group like a chloride or a methyl group to a little peptide tail that's coming out of that area, that will change how an immune cell reacts to an antigen it's just exposed to, whether it regards the antigen as self or foreign. A single little tiny change, chemical change, that's part of the epigenetic system. What I'm trying to kind of get across is how simple the system is in its operation, but in its effect, it's really major. It's a really very, very important part of it, and it's capable of taking signals from the environment.
Okay. And one thing, of course, that the public always wants to know is cures. When are we going to have a cure for these things? However, given the fact that it turns out that HIV and cancer and stuff are much more complex than we previously thought, do you think, therefore, that we were overly optimistic in thinking that we were going to have a vaccine for AIDS or a vaccine for cancer? But what are your thoughts?
Well, I don't think there can be a vaccine for cancer because it's too complex. All the different cancers are different, the causes are different, and so on. But I think that if you look at it now, patients with AIDS are living for much longer than they did. In fact, it's becoming a chronic disease, isn't it, with people on multiple triple therapy and so on for life. So the future for the AIDS patients is vastly better than it used to be. I think in cancer, we still don't know what fundamentally triggers it. Why you have cells that are, what mutations are occurring in cells that seem to be very specifically triggered. We know certain instances we do understand. For example, I'm sure that you're aware that sorry, I'm picking on something here, that we know that, for example, Angelina Jolie,
the movie star.
Yes, she's actually gone out of her way to act as a kind of representative to women to help people dealing with breast cancer.
Right.
She has a mutated BRCA1 gene. Think BR, breast cancer 1 gene. Now, that gene is a strange gene because it's a very large gene. It makes a very big protein, much bigger than usual. But what the protein actually does is it corrects mutations in other cells. So when the mutation takes place, we have the genome actually has the possibility of correcting mutations when they arise, and that's one of the proteins that does it. So what happens in the ladies who are unfortunate enough to have this mutation is mutations are arising in the breast cells, mutations arising in the ovaries, and this gene isn't there to correct them. So you can see how there's a cumulative effect now going on over years as more and more mutations are arising, and eventually they'll be sufficient, and it gives rise to cancer. So the patients are subjected to heroic surgery and all that now. And, of course, this, again, could be treated with pre-implantation genetic diagnosis. So you could eradicate it from a family pedigree in that way, too. But you can see what's happening, and that tells us something about cancer. That it isn't due to a single gene being mutated, usually. It's due to a series of mutations accumulating in the cells until they reach kind of critical mass where the cell metabolism is completely transformed. The cell no longer recognizes itself as self with all the other cells around it, begins to invade around it. And the other thing is retroviruses get called in, and they can even make the cancer cell become invisible. If the virus is expressed on the cell surface, it can make the cell invisible to the immune system. And so you can see a whole series of factors coming in that play an important role. But the thing is, we are recognizing we're coming to know much more about this. I'm sure that the future for cancer therapy, in 10 years' time, the future for cancer therapy will be a lot better than it is today.
And speaking about the next 10 years, given the incredible rate at which things are moving, you mentioned the CRISPR technology, for example, right?
Yeah.
If you had a crystal ball, of course it's always dangerous to make predictions, but do you have any thoughts about where things are going, given the enormous breakthroughs?
The really fastest thing is that, sorry to harp on this, is the epigenetic research. Because the epigenetic system allows us to put something in from outside that changes the expression of genes. There's a massive amount of research, more than 5,000 research papers a year. Some of these are coming across my screen, and I can't possibly keep up with them. And they're talking about this epigenetic system is certain that cancer, this thing is very critical to that. This is a critical thing for how a patient responds to a certain, say, anesthetic drug and so on and so forth. And that the research on that is taking place at an extraordinary rate. I think most of the big pharmaceutical companies are involved with it. So I'm expecting probably more breakthroughs coming from that side than specific about genes, because it's so hard to change a gene in, say, an organ or in a tissue. Even that is happening, because they've done studies in Duchenne muscular dystrophy, for example. Dogs get Duchenne muscular dystrophy just like humans. And so they've done studies in dogs where they inject a gene therapy into one leg, thigh of the dog, and a control into the other leg, and then they watch what's happening in the two legs. And they've actually shown some gene therapy improvement in the legs of the dog, and also by giving this gene therapy intravenously. So, this type of therapy, even for diseases of organs and tissues in fully developed adults, I think it is going to come in. And I suspect I think it'll be both. It'll be both genetic type research and epigenetic research, because they're both very closely related.
Okay, and any parting thoughts for people who may want to pick up a copy of your book, The Mysterious World of the Human Genome? Any parting thoughts?
Well, we've had very good reports from readers. The book is deliberately made simple enough for someone who is a non-scientist to understand their own genome. I hope they'll enjoy it. It's fun. I developed the railway analogy of DNA and chromosomes, where people take an ultramicroscopic steam train, ride along the chromosomes and get out and have a look. So I adopt a kind of fun aspect, so it isn't too heavy. So if they do pick it up, I very much hope they enjoy it.
Okay, well, thank you so much for being on Exploration.
Thank you.
Well, I'm afraid that's it for the second half of Exploration. Once again, our special guest today was one of the world's leading authorities on viruses and the human genome. He's the author of a number of great books, including The Mysterious World of the Human Genome, also Coevolution, about the evolution of viruses, and also Darwin's Blind Spot, the role of living interactions in evolution. And he's a consulting physician at the University of Sheffield in the United Kingdom, where we were able to track him down. And also let me say that we are just on the beginning steps of beginning to understand the human genome and the genetics of life itself. And let me just say a few things about some of the things that we didn't have time to touch upon. First of all is the question of the genetics of diseases. That's right, we can not only clone and understand our genome, but we can also begin to dissect the family tree, the genetic tree of diseases, and begin to understand where they are, where they hide out, and eventually how to cure them. Because once we know the genome of diseases, we know their weak spots. And as a consequence, new therapies, new drugs, and new ways of combating diseases arises as a consequence. Second of all, new cures for human diseases. We mentioned AIDS and we mentioned cancer. Even though we cannot cure them, we understand how complex they are, and we're beginning to understand new avenues of beginning to attack them. For example, cancer now, we're beginning to attack at the genetic level, at the level of genes, which was once considered to be basically impossible. Also, we're beginning to create new ways of repairing organs of the body. In the future, I think we'll have a human body shop. Already, we can create skin, bone, cartilage, heart valves, blood vessels, even complete windpipes and bladders, noses and ears, by taking tissue samples and growing them in the laboratory. And so that'll create a human body shop.